Bom dia a todos e todas. É um prazer estar com vocês aqui mais uma vez nessa edição de novembro do SBV-Talks, né? Lembrando que essa é a última edição do SBV-Talks do ano de 2021. Nós retornaremos, né, às novas apresentações a partir do mês de março de 2022. Né? E hoje não poderia ser melhor, eu acho que como encerramento dessa atividade que a SBV propôs a fazer mensalmente. Nós temos, então, um convidado extremamente importante, né, que é uma dos pesquisadores, dos cientistas mais influentes no ramo de pesquisa, trabalhando com vírus de morcegos. Eu, então, tenho o prazer de convidar o Dr. LinFa Wang, and then I'll switch to English now to introduce him properly. I have to write it down, so because I wouldn't be able to remember everything.
Yeah, sure.
So, if I may, Dr. LinFa, thank you very much for being here with us. Dr. LinFa Wang is a full professor and former director of the program in Emerging Infectious Diseases at Duke-NUS Medical School in Singapore. He has earned his PhD in biochemistry at the University of California, Davis, in the US. And after that, he has worked as a professor in research in many prestigious institutes around the globe, including the Cyril Australian Animal Health Laboratory. I don't know if I have pronounced that correctly, yeah. Dr. Wang. Yeah. And in Australia, and the Monash University in Clayton, Australia, and also at the East China Normal University in Shanghai. He has been awarded important prizes, and I'm going to mention just a few of them in his because of his research excellency, including the Eureka Prize for Infectious Disease Research in 2014, and many other international awards. He's an honorary professor at the University of Melbourne, in Australia, at the Wuhan Institute of Virology, the Chinese Academy of Science, among other past and present positions. Needless to say, he has published more than 300 papers, many of them paramount to the study of virus from bats. There was an interesting editorial from Science, and they nicknamed Dr. Wang the Batman. Am I right, Dr. Wang?
Yes, yes.
So therefore, I would like to invite Dr. Wang to thank Dr. Wang very much to be here and present to us at the Brazilian Society of Virology. I should mention it's quite late in Singapore. It's actually 9 p.m. right now, and he was very kind to be here with us. So Dr. Wang, thank you very much once more. We're very eager to hear your presentation.
Thank you, Professor Fabio, and all the committee members. As I said, it's a great pleasure. It's a pity that I cannot be there in person because I have never been to Brazil yet, but it's a warm-up. At least, we get to see each other on screen. But I do hope that in the near future, we can see in person. So let me know that you can see the screen properly now. Yeah.
Okay. Yes, we can see.
All right.
Thank you.
Okay, so the topic is bat viruses and the pandemic preparedness, and it's a pretty hot topic right now. So as I was telling Flavio, that we cover a few, pretty diverse topics. First is to give you a review of bat viruses, and then why bats? Why bats are such a good reservoir? And then the top topic of the origin of SARS-CoV-2. There's a lot of politics, but I said let's focus on the science. And then, although we're still in SARS-CoV-2 pandemic, but people like myself is already looked beyond to say what SARS-3 will look like. And then, why the current vaccine is not working as good. We're still in the middle of pandemic, and we still could not see the, end of it. So last topic is that my current research is try to develop a vaccine strategy that as a booster vaccine that can not only fight the ongoing SARS-CoV-2, all the variants, but also preventing the global community from maybe SARS-3 in future. Very ambitious, I have to say. So similarities between viruses and terrorists, right? So if you still remember, Donald Rumsfeld, the Defense Secretary, U.S., early 2002, basically made this statement. At that time, people laughed at him, but it's very philosophical. Fighting terrorists, he said there are three categories: the known knowns, the known unknowns, and the unknown unknowns. Obviously, the degree of difficulty dealing with is very different. You think of viruses, they're the same. They hide in the dark, but when they came out, they have, basically very, very dramatic impact on our society. So virus also come in three forms: the known knowns, the known unknown, and unknown unknowns. So this is what I have been doing bat-borne virus for exactly 25 years when COVID-19 started in 2019. And so I was involved in the discovery of the Hendra virus, in Australia, and that led to the Nipah virus. And I led the international consortium to discover the origin of SARS-CoV-1 from bats. And then I also involved in MERS and Ebola, and now I'm heavily involved in COVID-19. But when you think of these viruses, right, when Hendra virus first emerged, we never knew that paramyxovirus can be so deadly. So that was an unknown. Same for 2002, 2003, when SARS-CoV-1 emerged. Again, coronavirus was not known to be lethal for human, and yet SARS was quite lethal, case fatality 10%. So then when MERS emerged, then become a known unknown. So COVID-19 definitely is a known unknown. So in that regard, I always say, people like myself fear that, we did not do a good job. But the international community had 17 years to prepare for this known unknown, and still we did not do a good job to prevent it, or at least to mitigate. So that's 25 years in terms of the time span. But if you look at the geographic locations, the first virus we discovered, called the Hendra virus, started in Australia. And then that led to the discovery of the Nipah virus, where I am now, Malaysia and Singapore. And that, led to the SARS coronavirus was in southern China, so still in Asia. And then it moved to Middle East. We had the MERS coronavirus, and West Africa, we had the Ebola, and then COVID-19 back in Asia, in Wuhan. So as you can see, anywhere really can have major pandemic. And although nothing has happened where you are, but you had a pretty bad Zika virus, right? So that really, I'm talking about this bat-borne virus, but of course there are other viruses can emerge. So really started, in 1994, 95, we discovered the Hendra virus, and the first evidence of bats being an important reservoir is in 96. We had a serologic evidence for the presence of, at that time, it's called the equine mobility virus, and later changed to Hendra virus. So this is where 1996 looked like. If you search PubMed for bats and viruses, you get maybe 5-6 papers a year. And now, last year we had around 700, and this year is still going up. So the whole field of bat-borne virus obviously, has moved on, right? And then I still remember, about 12, 13 years ago. So this is the 10th anniversary for the discovery of the Nipah virus. So we had a Nipah colloquium in the University of Malaya in Malaysia. And we start to discover the topic, why we keep discovering virus from bats are so lethal. So are bats special as a viral reservoir? And I clearly remember that we have two camps. I led the yes camp. So I was trained as a biochem cell biologist. So I come from the genetics and immunology. I think there's something unique about bats. And in the other camp, we have, Dr. Peter Daszak, who is now the CEO of the Eco Health Alliance. He is an ecologist by training, and he's in the no camp. He thinks it's just the behavior and the ecological difference of bats. That's why they carry more viruses. But 10 years later, Peter Daszak, in the photo, that's him. So it took him 10 years to do the project and 2 years to get the paper published in Nature. The conclusion basically is we demonstrate that bats harbors a significant high proportion of zoonotic virus than all other mammalian orders. And when paper like this comes, the scientific journalists follow this very, very closely. So this is a Kai, from Science published this commentary, very long commentary. It's 4 pages, basically titled "Bats Really Do Harbor More Dangerous Virus Than Other Species." And he interviewed me and Peter. So he quoted me to say, this is a very important piece of work for anyone interested in zoonotic spillover events. And he quoted Peter Daszak says, meanwhile was gracious in defeat, says LinFa was right all along. So we are friends and competitors, right? So this really opened the question, of why bats. So the first debate is, are bats special? So this Nature paper basically concluded to say, yes, bats are different. Why? How they're different? So this has been my focus for the last 15 years of research. I go from Professor Flavio introduced me as the Batman. I used to correct people. I say I'm only a bat virus man, but because I don't do bat research until maybe 2007, I started to do bat research to really ask the question of why bats. So this is a long talk itself, but I'm just going to go spend a few minutes to say bats are very unique, as the only flying mammal. So I just give you some bat facts, right? During flight, bats' metabolic rate can go to 30-fold of their resting rate. Their heartbeat goes to about 1,000 beats per minute, and body temperature go all the way to 42 degrees C. Any of this happen to us, as in our human body, we'll be dead. We cannot handle this kind of stress. So obviously, bats has diverged from land mammal around 65 to 70 million years ago. So they had that 60 to 70 million years adaptation to make the modern bats. And so what we have learned, the lessons from bats as a host of viruses, we just had a pretty extensive review published in Nature last year. So if anybody's interested, you can read the paper. I'm not going to go great detail to summarize this, just to say that we believe it's that bats can do better in terms of host defense and the tolerance balance. We all know that our immune system is essential, right? That's the defense we need. But many of the diseases we have is due to over defense or over response, over immunopathology. So I don't know about your society. We have a cliche among the virologists to say very few virus kills us, we kill ourselves. So this is the diagram for the Nature review. Basically, we say bats kind of have two different from us, human or mouse. On one hand, they have raised base level, a higher base level of certain defense genes, like the interferons, the ISGs, heat shock proteins, and so on. Equally important is they dampen the overreaction of certain genes, especially in the inflammation pathways. So I think, bats now in my lab are used as a model to not only study infection, but bats also is an excellent model to study longevity, aging, cancer, metabolic disease, autoimmune disease, cardiology, and you name it. So if you're interested, we published a short viewpoint recently in the Journal of External Medicine. And so that just came out not too long ago. Okay, so now I'm going to switch to the topic of virus origin. It's pretty hot for SARS-CoV-2. And I have been getting lots of interviews on this. So I just give you, to say that the one of the most high profile one was organized by John Cohen from Science, another science journalist. So we had a whole hour, basically, four scientists debate about the lab leaker versus natural origin. And it's all very civilized and all scientific debate, Obviously, I have been working in this area for a long time and it has been, interviewed many times. But here, I just try to bring you back to learn the lessons from the first SARS-CoV virus. And now I call it SARS-CoV-1. So in 2003, August, for this young virologist in the audience, that's like 18 years ago. And the SARS-1 started in November 2002. And by early July, WHO declared the pandemic is over. So, but even then, we still did not know where this virus came from and why it infected so many people and suddenly disappeared. So the WHO organized this mission to really investigate the origin of the virus and the early transmission event, just like the mission early this year to Wuhan to investigate the COVID-19 origin. So I was fortunate enough to be one of the 8 international scientists, in that mission, and I was the only one lab-based. The others are epidemiologists, zoologists, and vets. They are more kind of field-based. But one thing I really to emphasize, we had Ann Shuka, Dr. Ann Shuka from American CDC, and who now is the deputy director. So I have to say the politic environment was so different. 18 years ago, we had a WHO mission, and we have a very high-profile American scientists in the team, and we got free access to every institute, including the Chinese Military Institute of Infectious Diseases. And then from there, we had a collaboration of scientists from 3 nations, again, USA, China, and Australia, and the 5 institutes. I led this consortium. And about 18 months later, we discovered that, a SARS-like virus is in bats. And so we declared that bats are most likely the natural reservoir of this class of viruses. So that's the first paper, basically. What I want to emphasize is that in that 2005 science paper is that we discovered 3 genomes that had the sequence identity around 88 to 92 to the human SARS-CoV-1 virus, okay? And then it took us another 8 years, especially led by the Professor Shizhen Li from the Wuhan Institute of Virology, that eventually we were able to isolate a live virus. Now the live virus not only kind of can use human ACE2, but also is closer to the SARS-1 sequence. So the overall genome sequence is 95% and 96% identity in the key receptor binding domain. But it took 10 years. So I have to emphasize that, right? 10 years later, we found a virus which is 95% identical to the outbreak strain of SARS-CoV-1. And the international community basically, believed the science and basically said the virus originated from nature. And now fast forward, right, for SARS-CoV-2 origin, there's lots of questions. As I said, I have been interviewed many times. So I'm not going to go through all the questions today, just give you the answers, right? Was SARS-CoV-2 a man-made virus? No. As far as I'm concerned, there's not possible to make that virus. Could SARS-CoV-2 come from an accidental lab leak? Highly unlikely, because you can only leak a virus when you already have isolated and cultured. Is it too late to search for the origin of the virus more than now almost 2 years? No, because all virus origin studies are retrospective, and I will give you a table. Can the geographic location of virus origin be far away from initial human outbreaks? Yes, and there are examples. So what are the alternative hypotheses for the origin other than lab leak and natural, Obviously, the natural origin can come in many different forms, and there are many possibilities, and I'm not going to, don't have the time to discuss today. So I just want to, give you the time frame, right? What kind of time frame we need to get the origin of the virus. So I tried to, you're all virologists, so I thought it's important to differentiate between the virus we're talking about. The virus we have the sequence now from humans are what I call is the outbreak virus, right? That's the virus first detected in Wuhan, but now we have 1,000 variants, and we have the variants of concern, 4 of them. And then we have the progenitor virus, which is supposed to be 99% identical and just the immediate sort of predecessor. And then we have the virus that are in the same class, but an ancestral virus. So we're talking about 3 different viruses. And then in terms of the origin, we're really talking about the progenitor virus and ancestral viruses. So now these are the 6 viruses, backbone viruses I have introduced in the early slides. And as you can see, the Hendra virus, and the Nipah, SARS, I was involved. And now I'm working on the COVID-19. Hendra virus took us 2 years, and that was a world record already in 94. You have the virus outbreak in human, and then 96, we discovered the origin. Nipah was faster because Nipah was closely related to Hendra, and they used our knowledge in the reagents. SARS-CoV, again, it's a record. Took 2 years to find the reservoir of the ancestral virus, and 5 months to discover the progenitor virus. And MERS-CoV, again, very fast, 1 to 2 years, and took 1 year to find that it's a camel. And now SARS-CoV-2, it took the scientists in Wuhan 5 days to find a progenitor virus in bats. But in terms of the the ancestral virus. In terms of progenitor virus, and whether there's an intermediate host animal X, it's a still question mark. Then you go to the other extreme, right? So we had the marble outbreak in 67, Ebola outbreak in 76. Look at that. 50 years later, we're still debating, for Ebola virus. For marble, we have stronger evidence, but Ebola is still being debated. So that can basically, origin discovery research varies from few years to few decades. Okay, so the reason that the Wuhan Institute of Virology was able to do that is because they have been searching and surveillance of bats for the last 18 years. So in their lab, they have already a database of sequence. So that was able to find the one virus. Now it's very famous, called the RAG13. It's a bat-borne coronavirus. It's basically very close related to human. And remember, I emphasized, it took us 10 years to find a virus which was 95% identical at the genome level. And now it took them 5 days to find a virus which was 96% identical, the whole genome to SARS-CoV-2. And yet, the public believe that SARS-CoV-1 has a natural origin, and then they suspect that the SARS-CoV-2 may have a lab origin. So that kind of itself really does not make sense, right? We have a virus much closer to a natural virus. So can SARS-CoV-2, be from outside of Wuhan or even outside from China? Now that's scientifically definitely yes, So I just give an example. I went to California, did my PhD. I arrived in San Francisco '82. So it's right in the early stage of the HIV kind of outbreak. I mean, as far as human outbreak is concerned, we first detected, outbreaks in New York, San Francisco in the early '80s, right? But is that a USA virus? Definitely not. But it took, almost half a century now. We have all the data to suggest that, it's clearly that it's a virus that, as early as the 1920s, that in Africa and is in non-human primates. So in terms of the SARS-CoV-2, where we should put our attention to, So this was a commentary published in The Economist in July 2020. So that's like, 16, 17 months ago. And the title is The Hunt for the Origin of SARS-CoV-2 Will Look Beyond China. Why? Here's why. Here is the map of Asia. So China is here and Wuhan is here. The darker the color, the pinker color means the greater the species diversity of bats. So Wuhan, of course, have bats, but it's not well known for diversity. Come to southern China, like Yunnan province, you have more bats. You go even further south to Southeast Asia and to where I live now in Malaysia, Singapore, you have more bats. So first of all, you have much high bat diversity in this region, southern Southeast Asia. But equally important is that within the border of China, in the last 20 years, there have been very, very intensive surveillance programs. So basically, you have a more species diversity in this area, and the surveillance effort is maybe 10% or 5% of what's within China. So if you put these two facts together, then I would say you have much higher chance to find any kind of SARS-related virus in this part of the world. Okay. Of course, it's difficult, right? There's so much travel ban going on and the lockdowns in different nations. So we cannot do the international collaboration that I used to do. But we still managed to finish one study last year, and it's with a group in Thailand, Dr. Supapan's group, and she has been surveyed back for a long time. So last year in July, she had one chance to go to an artificial cave and search the sample of bats, 100 of them, and then we collaborated. So it's published, and I'm going to go through very quickly. Just to summarize, we have both molecular evidence and serological evidence of SARS-CoV-2 related virus circulating in bats and the pangolins in this part of the world. Now, for any virologists in the audience, nowadays you read a paper, the first thing you go to is the phylogeny like this, the whole genome. Then you say, okay, Professor Wang, your virus, is this RACCS203. It's not that close to the Wuhan virus. The Wuhan virus is here. And yes, it's only 91% identical. Regi 13 is 96. So it's not the progenitor virus. But the point I try to make is that it's very easy to find the related virus, means there's a lot of related virus circulating in that part of the world. Secondly, is serology. So I want to pay attention, your attention, to this. So this is the virus discovered in bats in Thailand, and this the next one, the closer one, is IM102, is a virus sequence discovered in bats in Yunnan province in China. When we did the serology, I don't have the time to describe it. We have a surrogate virus neutralization test. We publish it in Nature Biotech if you're interested. So this serology test basically can do very specific virus neutralization tests without using live virus, and we can do in BSA2 labs. And only need a few microliter of serum. So we can do that with human, bats, pangolins, and anything. So people have used that to make for mink and the white-tailed deer in USA. The point I try to make is that we detect a very high neutralization, 95% neutralize the SARS-CoV-2 virus. And these are bat serum. I can tell you, I have been in this business for the last 30 years, and bats don't develop high affinity antibodies. So we concluded that this serum, this bat serum neutralized the SARS-CoV-2 virus, cannot be from that particular virus, the genetic sequence. So what we try to emphasize is that there is more than one virus circulating in that bat colony. And the way we prove that is we raised antibody. In our lab, we already have antibodies against this YN02. And this virus basically is identical, the receptor binding domain, to the RCS203. So as you can see that, we got the virus bat serum from Thailand, neutralized the virus by 95%. These are rabbit hyperimmune serum, so they should be much better than the infected bat serum. And yet, Regi 13 is close and get that level. The IM102 only get to 70%. So I think we are very confident there must be virus circulating in Southeast Asia much closer related to SARS-CoV-2. So this is what we discovered, and we predicted, and we discussed in our Nature Communication paper. Just 2 months ago, this paper now is in preprint and is being reviewed in Nature, I believe. So this is a study from Pasteur Institute in collaboration with scientists in the Laos. Okay, so this is much closer, to southern China. So this is very exciting because if you look at the coronavirus, many of you have seen this many, many times. So this is the image, the spike protein on the surface is the most important, and here is the green. But in the spike protein, as we know, the most critical part is what we call the receptor binding domain. It's a small kind of 250 amino acid fragment. So this study that just being reviewed in the preprint is that if you zoom into the receptor binding domain and you build a phylogeny, and now you can see, so this is the human sequence, and these are the 3 bat sequences from Laos, and now this is the Regi 13. So Regi 13 is distantly related, and these 3 viruses is much more close related. So how close related? They only differ by 1 to 2 amino acid. So it's basically almost identical. Now, consider the beta variant, from South Africa has 3 amino acid difference and a key difference which changes the binding to the ACE2. So we have a natural bat virus in bats in Laos has a IBD which is closer to the human SARS-CoV-2. So as far as I'm concerned, we already find the natural origin of the most important functional domain of SARS-CoV-2. More importantly, they were able to isolate a live virus. Because Regi 13, our sequence from the Thai bats, these are just sequences. We failed to isolate live virus. The scientists in Laos now isolate live virus, and the live virus can infect the human cells and be neutralized by a COVID-19 serum. Now you look at these, right? As I said, I have to emphasize again, the Wuhan outbreak started in December 2019. We're less than 2 years, and we already find so many SARS-CoV-2 related coronavirus in bats, all the way from Japan to East China, South China, now Laos, Thailand, Cambodia. Imagine once the border opens, if you do large-scale intensive surveillance in this area, you will have many, many more viruses to be discovered. And I believe that we can find a much close related to the progenitor virus. And it's a huge geographic range. The direct distance is 5,000 kilometers. Okay, SARS-CoV-3. So why we want to talk about SARS-CoV-3, we're even, far away from, get out of the SARS-CoV-2 pandemic. I think it's important because that's the lesson, right? If we did that after SARS-CoV-1, we may be in a much better place than we are now. So we had to look beyond SARS-CoV-2. I have been, doing this kind of virus for a long time, and I came to Singapore in 2012 July, and then we had a MERS in September. So 2013, the journalist from the Straits Times interviewed me, basically asked me to predict. And I'm a very conservative scientist. Usually I don't like to predict, but for this, I was so sure. I said, I'm almost certain that in the next 10 years, a new killer virus spread by bats will emerge. And you know what? I was right. But unfortunately, in my profession, if my prediction was right, people will suffer, right? So we had the COVID-19. So that, within that was 2013, right? So in basically 6 years, we got this outbreak. So, and it's not a fluke because we have been predicting this for a long, long time. And I have grants, the title is Combating the Next SARS and the MERS-like Viruses. And in 2019, I published a review in the Current Opinions in Virology. And that paper was out, just a few months before the first case of COVID-19 in Wuhan. And, we predicted to say that among the known unknowns, that coronavirus may be the most likely cause for future spillover into human, right? So that paper was went online in January. So just, 11 months before COVID-19. Okay. All right. So if you look at the coronavirus family, right? So unfortunately, the genus will also use the same alphabet. So we have the genus Alpha, Beta, Delta, and the Gamma. And we all know that Beta is the most dangerous because the 3 emergent zoonotic coronavirus all come from that genus. So we had the SARS-1, we had the SARS-2, and we had the MERS coronavirus. But there's a difference. SARS-1 and SARS-2 belongs to this subgenus called the Sarbecovirus. Sarbecoviruses are the SARS-related beta coronavirus, and they use ACE2 as receptor. And we know now these viruses are much more dangerous because they can, have much higher transmission among humans. Okay, so I think you can debate about this, but most scientists believe that if we're going to have a SARS-CoV-3, it will most be likely another Sarbecovirus, and it most likely will come from Southeast Asia again. So that's why, where we are now, I think we have a lot to catch. So why SARS-CoV-3? And in the context of the current, outbreak, because the current pandemic taught us a very, very, painful lesson. That means that the immunity, the protective immunity of coronavirus is very, very specific and short-lived. That's why we're suffering from reinfection and vaccine breakthrough infections right now. So I'm going to share 2 stories. Were published, you guys know already, but I just want to contrast, the Israel story. Israel was the best vaccinated and still remain the best vaccinated. So they will reach 62% already by June, and they only used the Pfizer mRNA vaccine, and they got very good real protection data. It's 94.3, phase 4 basically. But that did not last. until June, that's okay. From June to July, that protection dropped to 64%. And then September, they dropped to 47%, and the government immediately started this booster vaccine. So the lesson basically said it does not last as long as we wish or expected. Now the South African story. They have suffered 3 waves of COVID-19 infection, and by 3 different viruses. The first wave was the D614G. We basically call it the wild type or the parental strain. And then they had the beta, and then they have the delta. So what that says is that, if you had a beta, the population still is not sufficient to prevent delta. Okay, so now we know that protect immunity, the triangular relationship is not equilateral. So the parental strain, if you infect with this, then your protection against the beta and the delta is reduced by same kind of a degree. Around neutralization titer is around 1 to 3-fold. But if you are infected by beta or delta, and then the difference is even greater. So their neutralization titer can vary 6, 7 to 10-fold. So that's why that, remember that when we see this vaccine breakthrough, many people said, why don't we just build a beta vaccine? Fortunately, we don't have a large-scale vaccination use beta to replace the parental strain, because if you vaccinate with beta, the breakthrough by delta is worse than the first-generation vaccine. Okay, so the protective immunity is very virus and strain specific. So how do you measure protective immunity? There's a lot of debate, whether it's the T cells, a neutral antibody. My standard answer is both arms are important, T cell and the neutral antibody, but neutral antibody is the first line defense, and also neutral antibody is the one you can only practically measure on large scale. So this is a very milestone paper in Nature Medicine, published by Australian group. They basically, use data from 7 vaccine trials and from, outbreaks to come to the conclusion to say neutral antibody levels are highly predictive of immune protection from SARS-CoV-2. And many of you have read that paper. So they defined the neutral antibody level from, convalescent sera as a 1. And then the vaccine, you have, mRNA vaccine is high and the inactive vaccine is lower. Okay, so then how do you do a neutralization test? Because SARS-CoV-2 is a DSS3 live virus, you have to do in a biocontainment. And but the principle is simple, right? You mix your sera with the virus, and if you have a neutral antibody, you block the entry, and then that's no CPE. Otherwise, you have CPE. Okay, so this is a conventional neutralizing antibody, but requires BSF4. So we developed a surrogate virus neutralization test, and it's already published in Nature Biotech. Basically, it's a biochemical mimicry. So on the left is the live virus neutralization and the whole virus, and we use protein engineering to clone and express the ACE2 as a soluble protein. And for the virus, we clone and express the receptor binding domain, and these 2 IBD bind to ACE2. In the presence of neutral antibody, it's blocked. And then we conjugate the IBD with the HIP. So become a simple ELISA test, very, very quantitative. So this is X is the live virus neutralization, and the Y is the SBNT. The commercial kit is called the CPAS. As you can see, the correlation is very well. And we can differentiate between binding antibodies and neutral antibody. So in panel A, we have 4 binding monoclonal antibodies. So these are measured binding onto the IBD. And on the right panel B is the percentage inhibition. As you can see, 2 of the 4 maps, they had a very good binding, but they don't neutralize. So that's the difference between binding antibody and neutral antibody. And that's a species independent. We can use that for human, for mouse, for rabbit. And these are the species we already tried. And I don't have the time to add because this has just been used, as I said, in the white-tailed deer, So, and the kit was launched commercially available, and I think you're using, some people are using in Brazil already, and we got an FDA approval in November last year. Now we have moved on to develop the next generation of surrogate virus neutralization test. So this is a multiplex. So we made this modifications. Instead of coating the ACE2 on the solid phase, we put IBD onto the solid phase, and these are the magnetic beads. And the ACE2 is now in the liquid phase, and it's directly conjugated with the PE fluorescent. The principle is the same, right? In the presence of neutral antibodies, then you block that, and then you get this what we call in-tube competition, because we are now measuring neutral antibodies against different viruses, the different variants, and other subaqueous viruses. So this is the phylogenetic tree of the different subaqueous viruses, and the virus in bold are the one we selected. So this clade is we call the SARS-2 clade. So you have the alpha, beta, the pangolin virus, the Reg13, and this is the clade 1 virus. So we have SARS-1 and bat viruses, okay? And as you can see, their ACE2 binding is really, really very, very reliable, because the beta virus has the best affinity to ACE2, and it's reflected in our multiplex assay as well. Okay, so this is the data, So what we did is using the serum coming from the patients infected with the parental virus, beta virus, and we did this against the SARS-CoV-2, the white type virus, and then the 4 different VOCs. So as you can see from the parental virus, the best neutralization is against the white type, followed by alpha, delta, and the beta and the gamma are similar. If it's the beta, then it's gamma, alpha, white type, and the delta. And then if it's delta, the other way around. But as you can see already, the difference here is gradual. But the difference here, for example, beta to delta, and the delta to beta and the gamma is much more significant. So this is the thing I already told you, that it's not equilateral triangle, right? The beta and the delta are more distant related in terms of their protective immunity, And so we are crying out for a better vaccine. And John Cohen, the person who hosted the debate of the virus origin, also wrote a very comprehensive review in Science, and I encourage you guys to have a read. So he talks about DREAM vaccine, and I said DREAM comes in different generations. So if you look at the vaccine, just like the mobile phones, right? We go from 1G to 5G. The 1G is the vaccine we have. It used the original virus sequence. The 2G is people are working on now to say we want to use the VOCs. I'm working on the 3G. That's the pan-subaqueous virus. And then the 4G will be the pan-beta coronavirus. And then the 5G will be the pan-coronavirus, the whole family, okay? So I'm focused on this 3G pan-coronavirus. The reason is that I think we have the hope, because all these viruses use the same receptor. So I think we believe we can do it. And we made that discovery recently, and it's published in New England. So what we did is to go back to the SARS survivors in Singapore. They were infected with SARS-CoV-1 virus 17 years ago, and now they received a vaccine, the Pfizer vaccine for SARS-2. I want to ask, when you receive a vaccine, do you get a boost of neutral antibody against original virus? If they do we have now a broad pan-subaqueous virus neutral antibody in these patients that have been vaccinated against SARS-CoV-2? And this is the mechanism we were proposing. So if you have SARS, the immunodominant virus is very strain-specific, remember? So the SARS-1 specific neutral antibodies, the epitope is in circle, and the COVID-19 is in triangle. So you can do booster, and you can do many, many vaccination. If you use the same virus, you're only going to boost the immunodominant one, the original antigenic scene, right? Only when you cross boost, like we did in this patient cohorts, they infected with SARS, now vaccinated with SARS-2, you force them to boost these cross-neutralizing epitopes. And we achieved that. So this was published in the England Journal. And the journal was so nice. They made a beautiful, very, very professional animation. So I'm going to save my time, and I'm not going to just show you the video, and it's much more impressive.
SARS-CoV-1 and SARS-CoV-2 belong to the species SARS-related coronavirus and the subgenus Sarbecovirus, but are in two distinct phylogenetic clades. Emerging SARS-CoV-2 variants pose a challenge to the effectiveness of current vaccines. A vaccine capable of controlling current SARS-CoV-2 variants of concern, as well as preventing infection by other sarbecoviruses, would be optimal. To investigate the possibility of cross-clade boosting of neutralizing antibodies against sarbecoviruses, investigators obtained serum specimens from 8 long-term survivors of SARS-CoV-1 infection who had received the Pfizer-BioNTech mRNA SARS-CoV-2 vaccine. Surrogate virus neutralization tests were performed to analyze the specimens for neutralizing antibodies against 10 different sarbecoviruses, 7 from the SARS-CoV-2 clade and 3 from the SARS-CoV-1 clade, including variants that affect only animals. The authors examined 5 serum panels consisting of specimens from unvaccinated SARS-CoV-1 infection survivors, unvaccinated patients with SARS-CoV-2 infection, healthy vaccinated persons, vaccinated survivors of SARS-CoV-2 infection, and vaccinated survivors of SARS-CoV-1 infection. All vaccinated survivors of SARS-CoV-1 infection had high levels of neutralizing antibodies against both SARS-CoV-1 and SARS-CoV-2, including several variants of concern. These patients were also the only group with a broad spectrum of neutralizing antibodies against all 10 sarbecoviruses, including bat and pangolin viruses with the potential to cause human infection. In this study, high levels of neutralizing antibodies against a broad range of sarbecoviruses, including both SARS-CoV-1 and SARS-CoV-2, were detected in survivors of SARS-CoV-1 infection who were immunized with a SARS-CoV-2 mRNA vaccine. Further study is required to see whether other combinations of antigenic stimulation from different sarbecoviruses might be able to elicit broad immunity to the entire subgenus. Full study results are available at nejm.org.
All right, so that just gave me the pleasure to really thank my team. This is the Christmas party of December 2019, basically when the COVID-19 was just starting in Wuhan. Of course, we did not know. We were very happy and tried to have a relaxing 2020. But since then, we have been working very hard, no weekend, no night, no holidays, basically. But I think we have achieved a lot. And we have lots of collaborators, around the world, and I thank for them. And then just lastly, this demonstrates that dealing with pandemic, you need a One Health approach. So we want a bit. So I'm organizing the 7th World One Health Congress in Singapore, November next year. So exactly a year from now, and hopefully it will be face-to-face meeting. And I really welcome, many of you in the audience to come to Singapore. Thank you for your attention.
Hi, Dr. LinFa.
Yeah.
Thank you very much for your talk.
Yeah.
It's
Thank you.
talk.
Yeah.
And we are very impressed
Mm-hmm.
with all you show here for us. And
Yeah.
for us in Brazil, this is very important because how
Mm-hmm.
we talk before, we have a very, very large diversity. I think it's the biggest diversity of bats.
Yeah.
In the world is here in Brazil, and that is fantastic.
Yeah, no, I'd love to collaborate with you guys. Have the test, and then we can collaborate.
For sure. We have now everything, our email and phone calls.
Yeah. Mm-hmm.
You never more rest. Thank you very much. It's
sure.
Really, Flavio, please.
I actually, well, it was fantastic, Dr. Wang.
Thank you.
Really fantastic. And I have a question. I am a biologist. I'm not, a zoologist, so I don't understand much of bats. But I know that bats from the New World, they are quite different from bats from the Old World. You have like the Megachiroptera there in Asia.
Yeah.
And you have the Microchiroptera here in the Americas. So how does it correlate with coronavirus? Is there any study about a coronavirus from Microchiroptera from the New World?
Yeah, certainly that, so first of all, why bats? That's what I'm studying. The bat immune system definitely is unique. So that regardless of from kind of the old world or new world, the big and the small, because they are flying mammals. I think it's adaptation to fly. In terms of their ability to carry virus, then again, we still study there's a special affinity. Coronavirus loves bats. So you have coronavirus in bats in your country, in my country, anywhere in Australia, okay? But the SARS-related coronavirus is only carried by this genus called the Rhinoceros. So that's good news for you guys, because this bats does not exist in your part of the world, okay? So that's the good news. The bad news is now, that's why the One Health is important, right? We have the zoonosis, we call the forward zoonosis, animal to human transmission, but SARS-CoV-2 taught us a lesson. The reverse zoonosis is equally efficient, equally dangerous. So now the nightmare we were worried about has happened. We were worried about this virus now can infect the bats in your continent, because it's not the natural, because the rhinoceros bats does not exist. But there are bats have the ACE2 receptor, which is susceptible to SARS-CoV-2. So we have not detected it in bats yet, but we have detected it in wild white-tailed deer. Now in USA, you have seen the report. I mean, the seroprevalence is already 40%. And in terms of lineages, they have like half a dozen different SARS-CoV-2 easily detected in their deer population. So basically, we have a new reservoir for SARS-CoV-2 already. Okay.
That leads me, that if you allow me, can I monopolize a little bit more?
Yeah.
So it leads to my second question, because here we're starting to find similar, results. For instance, we have found people from different parts of the country have found antibodies.
Yeah.
I don't know if they are neutralizing or not, but they have found SARS-CoV-2 antibodies in other mammals, such as coatis, which is a small carnivore.
Yeah.
And also in wild carnivores, such as wild cats, and if I'm not mistaken. But in coatis, I'm quite sure it was found.
Oh, so here's a collaboration. I can send the kits to you, because my CPAS, the Surrogate virus neutralization test, is species independent. So the USDA used that test to do that deer study. So any species in your country, if you suspect that there's a spearback human to animal infection, my kits can tell you. It's a neutral antibody. It's a neutral antibody.
Yes, I'm absolutely excited, because I just did some ELISA, regular ELISAs, but they don't tell if it's neutralizing or not. And the amount of antibodies was quite high. But now with your kit, we can know if they are neutralizing or not.
Yeah, so and my kit, as I show you already, the multiplex platform can even differentiate between neutral antibodies of the parental strand, delta strand, pangolin strand, bat strand. So it's that high resolution. Yeah.
Great. So I'll hand over to you guys for all the questions.
Dr. LinFa, just completely, Flavio, this CPAS is only it's ELISA model or is Luminex model? I saw in your slide.
Two versions. The single-plex is an ELISA module. Okay. The multiplex is the Luminex, because you cannot do multiplex with ELISA.
Wonderful. And this is commercial
available? You have a distributor in Brazil. I can refer to you.
Flavio, let me ask more one question before you open for all the audiences. Brazil is the largest diverse of bats, and every time I ask my question for myself, like why all the pandemics start in Asia, or especially in China, and not in Brazil, and we looking a lot of bats for coronavirus, and what we found here is much more alpha coronavirus than beta coronavirus. Do you think that it's possible this alpha coronavirus can be next emerging virus, or cause a human disease, or can be a pandemic virus?
Pandemic is.
I know that it's just.
Pandemic is hard to predict. Just like SARS-CoV-2, I always say, even when 2013, I said there would be another bat killer virus spill over, but nobody predict that scale, right? Okay. But in terms of alpha virus spillover from bats to humans, definitely that can happen. So that's what I say. Before the seminar starts, I'm a strong believer of serology. So if you do your bat virus survey in Brazil, if you found an alpha coronavirus, and then if you do similar thing like what I'm doing, do a multiplex serology, and then you sample your human serum, I can bet you will find spillover into human already, because this spillover could be mild disease or no disease. Okay, so I can tell you, like a SARS-1, SARS-2, now, we and other bat-borne viruses, if you search hard enough, you will find the related virus jump into human more frequently than but we only pay attention when they have a cluster and a severe disease.
Yeah, of course.
Yeah.
Thank you very much. You clear my mind a lot. I think we are in the right direction, Elena. That is we're looking for.
Yeah.
Yeah, Dr. LinFa, we had a lot of questions here from the audience. Actually, it's a lot of compliments. Everybody saying that your talk was amazing, and we agree with that.
Yeah.
And we have one question about the end of your talk. About they ask actually if are there any mRNA vaccines against SARS-CoV-1?
Yeah.
And do you plan to vaccinate COVID survivors with it to test the cross-antigen stimulation? Can you?
Yeah, obvious question. Of course, I have been working on this since May, and we have many companies, and Gates Foundation. Everybody is interested in that new vaccine strategy. So my plan is going to be multiple platform. We will have mRNA vaccine, protein vaccine, viral vaccine, and we are not using just SARS-CoV-1. We use a consensus clade-1 sequence. So we already have a pattern, and but of course, in a pandemic, IP is less important. But in these days, university will ask you to have an IP. So we have an IP. But at the same time, I'm trying to develop this vaccine as fast as possible. So this will be what we call a 3G COVAX, the third generation coronavirus vaccine, and it can be only used for boosting, because it will be useless for primary, because it's not against COVID-19, it's against a clade-1 virus. So it's a SARS-1, human SARS-1, and bat SARS-1. And we already have demonstrated very good preclinical results in mouse. Yeah, so we are working very hard. If everything goes, according to plan, then sometime next year we will have a clinical trial. Yeah.
Thank you. Wonderful. Yeah, and I think I have a question as well about the virus surveillance in bats.
Yes.
Dr. Durigon touched, we have, and that becomes more frequently to do that virus surveillance. And my question for you is more, how can we target better, like you said about the serology, but sometimes what we have here, and it's very active, it's the rabies surveillance that they have already some bats. So we have plenty of tissues available that we could screen for virus.
Yeah.
So what will be your best strategy to find a virus or interesting or more pandemic or interest virus?
Yes. So for this kind of scenario, that you already have samples, from bats that harvest for other purpose, rabies or whatever. And then I think, very deep NGS is the best. What you want is that discovery-driven study rather than a hypothesis. Because to me, right now, the flavor of the year is coronavirus, right? But we know paramyxovirus, filovirus, real virus, all can jump out of bats and cause disease. So to me, is that it's a volume study. EcoHealth and USAID was trying to drive this global volume project. And I always say, South America is the country I think need more effort, because we have not done enough surveillance and we have not found enough virus in bats in where you are. So this is the first discovery. After discovery, then we do risk assessment. Okay. And that risk assessment can be done bioinformatically or epidemiologically, but I'm saying is serologically, because now the technology, we can build up multiplex assays, for 10, 20 different viruses. So once you've done the virus discovery using a next generation sequencing, you do a risk assessment, and then you do serology, because serology has advantage and disadvantage, because the advantage is actually can detect a cross-reactivity. So if we had an N protein for SARS-1, we would have detected SARS-2 already. All right. And that is also the disadvantage. People argue to say, oh, it's not specific, it's just background. I say, no, it's not background. Background is something that stick to your plate. This is a real antibody bind to your antigen. It's just cross-reactivity. If you're doing discovery, cross-reactivity is important. Of course, you need to have the next test, like the surrogate virus neutralization or pseudovirus, to do the more specific, confirmation later on. But the discovery part is next generation sequencing followed by multiplex binding antibody, and then confirm with neutral antibody. Yes, so that's kind of my strategy.
Thank you very much.
Thank you, Doctor. Oops.
Go
ahead, Liana.
Yeah. Do you know if there is any relation of the bats' feed habits with the host of any coronavirus? Because they have different habits, or even for cohabitation and what they will eat. And is there any relationship with the infection?
I mean, that's hard because, different coronavirus or different bats carries different coronaviruses. But to a large degree, the transmission is a sort of a fecal oral transmission, So that's the only thing you can think of, their behavior and the, environmental contamination. Otherwise, I think we have not found any bat behavior or bat species associated with certain coronaviruses yet.
Thank you. And we have another question from the audience. I'm not sure that I can understand what is written here. Simply, what's the best material to find the
virus in bats? So that's a very related question as we're discussing, right? So I gave you 3 examples, right? So for coronaviruses, as I said, if you're not allowed to sacrifice the bats, then fecal or anal swabs is the best, right? And then for paramyxovirus, like the Hendra and Nipah virus we studied, urine. So this is the best. What you did, what we did is that, of course, it's highly risky. You have to protect yourself. Bats go out nighttime and they come back. The first thing they do is they urinate. So we call this the urine shower of bats, right? But these are the liquid gold for virologists, So what we go there is we know when they come back, let's say 5 a.m. and 4:30 a.m., my PhD and a postdoc will go there with protective gear, put a clean plastic sheet underneath the bats, where, you already observe where they're going to put the, urine. And the urine shower happens, and then you collect the fresh urine, put into your virus transport media. The most productive job we got, one job, we isolate 4 different viruses, live viruses in Australia. Okay. Of course, you have to be lucky, right? So that's the second type. So the paramyxovirus and coronavirus, that's kind of lucky. We can do that without sacrifice. But for Ebola and Marburg, the filoviruses, CDC group discovered that the spleen is the tissue that most likely the virus is hiding. So that's difficult because that's invasive sampling. So the question to the answer to your question is it depends which virus you're targeting. Yeah.
Right. I think you go ahead.
Is there any other question? Because I'm just worried about the time you have to.
Don't worry about the time. I'm used to work nighttime because.
All right.
Yeah. Since COVID-19, my nighttime every night I have something on. Yeah. No, I enjoy this. I try to, as I said, I have never been to Brazil, and the chance to interact with Brazilian virologists, I just love it. Yeah. Thank you. Yeah.
Thank you. We don't have another question for the audience. I don't know, Professor Durigon, if he has any question.
Yeah.
Oh, thank you very much, Dr. LinFa. I have a million questions to talk to you, but I think we go slowly. And we are very exciting. I think you open a lot of opportunity for us to do this collaboration. We now in the middle of a big project that we are screening samples from bats from different areas in regions in Brazil, like Amazon's Northeast and extreme south of Brazil, and different biomes. And the serology, we collect the blood from these bats. The way that we are collecting now, we get just a little drop and put in the PBS and frozen. That is the way, because it's not easy, in the field to collect the blood first. And I think we are very exciting. If you have opportunity in the short future, in screening this serum with your CPAS multiplex test, because it will be fantastic for us.
Sure, yeah. So I just noticed Professor Hendra Kroon in the audience. She has been very, very helpful for the virology journal, and she's the champion for Brazilian virologists. So I just want to thank her, and say that's great.
Yeah, Hendra was my former mentor, and
I see.
She's a former president of the Brazilian Society of Virology as well.
Okay. Yeah. No, it's great. It's great. Yeah.
Thank you, Hendra.
Okay.
All right. So I just would like to say, Dr. LinFa, that sometimes you go to a presentation like that, and you think, "Wow, that's a nice presentation." Then you go home and forget about it. But sometimes, very few times, you go to a presentation where we came out with so many ideas, and this is one occasion. So thank you very much for your time, for your availability to be here with us today, and we're gonna work on that thing that we're never to Brazil yet, so we're gonna work on that and bring you at some time.
Okay.
All right.
As long as the travel ban lifts, I look forward to it. And then we can do that offline. I'm really interested to collaborate with you to get your wildlife serum. You're already seeing antibody to SARS-CoV-2. Let's confirm this is a neutral antibody, and then you just add a new species, because there's already 15, 16 species that infected with human SARS-CoV-2. So whatever you discover, will be interesting. Yeah.
Great.
Okay.
Well, with that, I'd like to say goodbye. Thank you very much once again. Switching to Portuguese now. Pessoal, muito obrigado. Espero que vocês tenham gostado. E então com isso a gente encerra, né, essa nossa última sessão, né, dos SBV-Talks de 2022. Professor Edson, professora Helena, se vocês quiserem falar mais alguma coisa. Não?
I just like to thank Dr. Wang for his kindness and to take the time to talk with us, even though we don't have a lot of people watching you now, but it will be available for our members, and they will enjoy your talk. It was really, really great, and thank you again for your time and to stay till night in Singapore.
No worries. Okay, thank you. Bye-bye.
Thank you. Bye-bye. Have a good night.
Bye.